Today, we are diving into the fascinating world of microbial preservation methods. Let's explore the 8 incredible techniques that scientists use to maintain these tiny organisms for extended periods. First up, we have the agar slant culture method. Microorganisms are inoculated on an agar slant, covered with sterile mineral oil, and stored in a refrigerator to provide anaerobic conditions and prevent dehydration. Next, we have refrigeration, where microorganisms are stored at 0 to 4 degrees Celsius to slow metabolic activity and prolong viability. The paraffin method involves pouring sterile liquid paraffin over cultures to create anaerobic conditions and prevent dehydration, allowing microorganisms to remain dormant for years. In the saline suspension method, bacteria are suspended in a 1% salt solution to inhibit growth and then transferred to agar slants when needed. Cryopreservation is a cutting-edge technique where microorganisms are frozen in liquid nitrogen at -196 degrees Celsius, preserving cell viability for long-term storage. Lyophilization, or freeze-drying, involves rapidly freezing cultures at -70 degrees Celsius, dehydrating them under vacuum, and storing them at 4 degrees Celsius to maintain viability for several years. Preservation at very low temperature involves suspending microorganisms in nutrient broth with glycerol, freezing them at -15 degrees Celsius to -30 degrees Celsius, or in liquid nitrogen to drastically slow down metabolic activities. Lastly, preservation by drying and refrigeration includes drying microorganisms over calcium chloride under vacuum, reducing moisture and metabolic activity for long-term stability. These methods are not only crucial for scientific research, but also showcase the incredible ways we can preserve microbial cultures for future studies. The world of microbiology is truly awe-inspiring, and these preservation techniques play a vital role in advancing our understanding of the microbial world. Thank you for joining me on this enlightening journey through the world of microbial preservation methods.